I give a warm welcome to the students of the University of Parma. Welcome here. And now adesso do la parola al professor Graziosi che continua un po' l'argomento di cui abbiamo parlato fino adesso e ci parlerà il professor Graziosi insieme al professor Pallavicini, sono del mio dipartimento, del provvedimento di Scienze della Vita dell'Università di Trieste. Ci parla di un argomento interessante. Voi sapete che Trieste è forse uno dei più importanti, se non il più importante porto per il caffè, no? In Europa, ce ne parleremo, in Italia di sicuro. Si parla della distinzione fra il caffè Arabica e Robusta, identificazione delle cultivar e tracciabilità biologica. Come vi ho anticipato prima, Professor Graziosi will speak in English because most of the students which are there are not Italian and so they need a talk in English.
Okay. Well, thank you. First of all, I want to say thank you to the organizer for giving this opportunity also to talk to the student of the Slow Food and another university. I'm going to talk about coffee and you got a real coffee about an hour ago. Now we are going for virtual coffee. And a very narrow topic, that is, we heard how to distinguish many species. Now we work only to two species. One is called Arabica, caffé Arabica. The other one is caffé Robusta. It's called Robusta. The scientific name is canephora. Now, what is the problem about coffee? The problem, you see a grove in the coffee bean. The coffee is produced in certain countries and consumed in different countries, mainly in the temperate belt of the world. So this poses a lot of problems also on research, and well, I'm not going into research. Another problem is that if you look at the square apple in the poster here, probably you can recognize a Granny Smith apple. But if you look to this bean, you can't say whether it is Arabica or canephora or Robusta or what it is. So this means it is very difficult to decide which species belong to the coffee. And the price of Arabica or Robusta can be very different. The price of Robusta is generally less than half compared to that of Arabica. The record of Arabica is maintained by the Luwak coffee coming from Indonesia, and a few months ago I saw it sold for 2,200 euros per kilogram. So the commercial interest is very high and important. Now also the interests of the producer are different. They are interested in specific agronomic traits, and while we are interested in the quality. Okay, to approach different interests, the consumers and producer, we develop a general approach to coffee that is genomics. We look at all the genes of coffee, and so we include in one type of analysis the interests of the consumers and the interests of the producers. Of course, today I'm going to talk about the interest of the consumer. In doing so, in doing this type of research, for instance, we classify many genes of caffé Arabica. Now we are working also on canephora, and this is an example of the function of 2,000 genes. But by now we have a collection of express sequence of DNA of 170,000 sequences, which is the largest collection in the world and comparable to a similar, same size produced by Brazil. Okay, the goals, as I mentioned, are to identify good agronomic traits and identification of quality characteristics. Here is an example also of differential expression of the genes we identify, but this is science. Let us go to what is more relevant for this meeting, and that is the identification of which type of coffee we have. So the first problem is distinguish between Arabica and Robusta. We developed some molecular tools very much in line with what was reported in the previous presentation. So we have specific pieces of DNA which are in the nucleus and other specific plastic DNA which allows us to identify the difference between the two species. Until now, the identification was carried on. I'm looking for different. Probably I didn't put it. Okay, it's not here. Oof. I may. What's happening? Yes. Okay. Sorry. Until now, the characterization, the distinction between Arabica and Robusta was done on two small metabolites, cafestol and caveol, which are two small molecules, and is made on the different quantities as reported in this slide. Unfortunately, there is a large diffusion, there is a recent large diffusion of hybrids between Arabica and Robusta, and this changes the ratio of these chemicals. So nowadays this test is very unreliable. It's not, if you run this test, you can't say whether you are in front of Arabica or Robusta coffee. While if we go to molecular biology, these white bars are pieces of DNA isolated in electrophoresis. And you can see that we have two bands for the three first samples, which are different varieties of Arabica, while we have only one band for the Coffea canephora, which is the Robusta, and also for another species of coffee which is not commercial, but is eugenioides. Why is that? Because Coffea Arabica is the only tetraploid species of coffee. So, and the progenitors, the ancestor of this species is canephora and very likely eugenioides. And they have small differences in the dimension of their original DNA. So Arabica must contain the two pieces of DNA coming from the progenitors. So it is obvious that Arabica must have two bands compared to canephora or eugenioides. Okay, this test works only on single beams. Okay, we can work either on green coffee, which is very easy, but we can work also on roasted coffee. It is not, if it is not being transformed into coal. If it is coal, it's very brown. We cannot work anymore. But in the majority of cases, we can work on that. Then comes the problem.